So we'll start at 2 and then we'll go as long as you guys have questions. But do you guys have any questions right now? You have. I'm very glad you have. Go ahead.
Not about this, but I have some other questions. The other day you talked about a microenvironment and acidification done by human cell. So we have this receptor on normal cell or human cell, carbonic anhydrase receptor, 12, especially membrane receptor. So by inhibition of this receptor, we can avoid extracellular acidification. So you haven't included it in when you're talking about different receptors. So I wanted to know your opinion on that.
Yeah, so I don't know about carbonic anhydrase receptor, but lactate acid, which is released, there are obviously those receptors there, and people have tried to inhibit that. So if you inhibit lactic acid secretion, in some way it is good because it prevents it from getting acidified, but that alone does not really change anything for cancer because they have so many alternative pathways. You shut down one, the others take over. So, and also they can also use lactic acid, convert back to pyruvate and feed it to this Krebs cycle there. So lactic acid inhibition or carbonic anhydrase inhibitor alone is not enough there. The problem is this: you cannot target everything which goes wrong in cancer because normal cells will also not survive. So you have to go for those guys which are, first of all, they should be ideally different between cancer and normal cells, right? So that only cancer cells are effective. The second very important thing, they should be the drivers. They are the one running the show. Lactic acid secretion is helping, but that is not running the show. You want to go to the culprits who are running the show, who are responsible for lactic acid formation and all this acidification of the environment. You want to go up and go to the master regulator. See, when you are even giving a combination therapy, right, it is considered very toxic because you are hitting two major pathways, right? But here, everything we are weighing against a fatal disease, so we are taking more and more risk, right? Because it's a question of life, right? So decreasing acidification is good because if you decrease acidification, you can let immune cells come close to it, right? But you know, cancer cells are employing so many ways. They are, you guys, some of you guys, I don't know who was telling me about these exosomes which are released. Somebody, you were telling me, right? So exosomes are really bad. So what are these? These are small vesicles. So cancer cells are releasing these exosomes and they will attack clean, nice neighboring cells who have nothing to do with cancer. And they will have all the bad things in there like RNA and different kinds of stuff, oncometabolites. They will go there, they will make them cancerous, right? So that is why, see, they are using immunotherapy. So immunotherapy is waking up the immune system. So it is going to attack in many different ways, like basically recruiting powerful enemies of tumor. But you know, you are right, you never know. We have to try everything. We have to try everything. Nothing is bad. Maybe using inhibitors of lactic acid in combination with something else is very good, right? So, and I'm sure it has been tested. I am 100% sure.
One molecule, SLC 0111, it is under clinical trial for carbonic anhydrase 9 inhibition because it is overexpressed in tumor cells.
See, so it is there. So we have to test it, and unless you test it, you really don't know what is going to work or not work. There are thousands of targets for cancer. People are testing.
Actually, before this conference, there were some Japanese scientists who came.
Yeah, I know, yeah.
So they, that sir It also has published one paper on photodynamic therapy for carbonic anhydrous.
That is becoming very popular these days, photodynamic therapy. Yeah, my friend at BITS Pilani, he's also working on that thing. So yeah, people are trying to treat cancer in 100 different ways, you know. So, and it is, you know, it is getting better day by day. Everything is adding up in our arsenal. So we can treat even aggressive cancers these days, right? So we are making huge progress. The only problem is that if cancer cells were not resistant, we would have eradicated the disease by now. Every time we treat them with a new drug, they come up with a new resistance mechanism. So right now in prostate cancer, the worst cancer is neuroendocrine prostate cancer. That did not exist before. This is man-created cancer. They come because we treated them with androgen receptor inhibitors or androgen signaling inhibitors. So that's a constant fight between man and cancer, you know. But I always think that one day we'll discover something so trivial and the cancer will be gone. Like, you know, you don't attack it at the cancer itself, you just switch off something else somewhere. It's like to remove the darkness. You, instead of, you know, removing the darkness and throwing darkness out in small pieces, you just turn on the light somewhere. You know, I don't know how, but I'm hoping that one day it will be a dream that this disease doesn't exist. Like, you know, we have eradicated thousands of diseases. Yeah.
I was not able to understand how cancer cells are getting resistant to radiation, because it essentially goes and damages the DNA every time you expose it to radiation. So how is the resistance? I mean, is it DNA repair in some sort?
Yeah, it is the upregulation of DNA repair enzymes. Radiation resistance is the most common thing. And you know, I had a student some time ago from China. He was a postdoc in my lab. So he's a prostate cancer oncologist and in Wuhan. So he actually works on this radiation therapy resistance. So in that he has shown that the metabolic pathways are separate, are different in radiation-induced cells versus non-radiation-induced cells, and the metabolites which are formed, they are the one which cause cancer. So when you give radiation therapy, DNA, upregulation of DNA repair enzyme, then the metabolism also changes. The enzymes are different between, you know, not in cancer cells versus radiation-induced normal cells. I mean radiation-treated cancer cells, and so these enzymes behave differently to become resistant. So there is, I was trying to tell you, I honestly cannot remember what is that, some CETSA or something, it is called CETSA or something. I will check it today and tell you tomorrow. So I know of that person, like I don't know him personally, but I know of him. So he has developed this CETSA thing, CETSA or something to that. So there what he does is that he basically takes something which will not even touch normal cells, and it will get disassembled within cancer cells, because cancer cells express an enzyme called alkaline phosphatase in huge amount, and he assembles this whole structure in such a way that if alkaline phosphatase is there, all you do is just pull one trigger and the whole thing will disassemble. Like it is like a whole brick of things going there, like just literally bricks like, and you just pull out just one piece like house of cards and the whole thing disassembles. So it's like giving, what we really want to do is a truckload of bricks on the cancer cells, damage them so badly. This is not molecular mechanism, this and that. You just basically hit them and you die. Don't get up again, right? So I'm hoping this kind of therapy will not lead to resistance, you know. So people are trying all different kinds of things. If I find it today, I will at least include one slide to show you what is that therapy, you know. So people are coming up with really innovative stuff. More questions? Yeah, go ahead.
Ma'am, as we discussed about the gut microbiota playing an important role in the cancer progression, so there are studies that the gut microbiota or the bacteria, they are translocating to the tumor site and remodeling the tumor microenvironment or the intratumoral microbial value. So are there any specific microbiota based studies? We discussed about FMT, but I want to know that are there any the specific therapies targeting the translocation mechanism or the intratumoral microbial profiling?
So what I know is that they recommend, so I don't think they are targeting microbiota itself in the tumor site, like they are not injecting it there, but what they recommend is to eat healthy probiotic kind of stuff so that you improve the microbiota in general. They are also doing the fecal thing. I don't know exactly how it does, but they are delivering the good microbiota that way also. So the whole idea is to systemically have the better microbiota in the entire body. I am not aware of that they are targeting microbiota directly in the tumor site. See, what I have understood from all of these things, that first of all, our knowledge is really incomplete because microbiota did not even exist 10-15 years ago. This is a new component. So there are so many components of this cancer disease alone that we are completely unaware of, right? So we have less than 1% knowledge, this is my thinking, about cancer, right? So based on every day the knowledge we are gaining, we are changing therapy. But what is miraculous is that, so before I come to the miraculous part, so like there are DNA methylases 1, 2, 3, 4, 5, whatever they are, right? So one is bad, you use inhibitor against one. Maybe one is also doing some good thing, but when you're inhibiting, you are also killing the good part of it, right? Same thing, or you cannot specifically inhibit DNMT1 because it has cross reactivity, right? So if this room is full of different problems, we are aware of 1% problem and we are trying to cure that problem. But what is miraculous is that, of course, when the cancer is there, we have to treat it. But the miraculous part is that if we are exercising, eating well, somehow that exercise, whatever the benefits are, they know exactly which one to inhibit, which one to activate. And we have zero knowledge. We don't know what is happening in the body. We are just exercising. And the body actually knows what to turn on, what to turn off. It takes care of every single thing, right? But again, once the disease is there, the body is the one which is actually fighting the disease off. The knowledge that we have gained is that, okay, immune system is shut off. We need to reactivate the immune system. Right? So it is extremely important that we gain the knowledge there. So coming back to your question of microbiota, the way they are treating is now systemically the whole entire the cross body. They're trying to improve the microbiota without going individually, because our knowledge is really incomplete in that area. There are trillions of microbes there. We really don't know which one is doing what. What we know is that microbiota is supporting tumor formation, and if it was good, there would no tumor formation. But we don't know at a molecular level which one is good, which one is bad. Is it making sense? Yeah. And also, many things are circulating in the blood when someone has cancer. So it's affecting other than tumor cells like metabolites, oncometabolites kind of things. I was talking to a friend, I don't even know who I was talking to. So he told me a very funny thing. He said if you measure the level, yeah, I remember whom I was talking to. He's a person of Indian origin who lives in US. And he said that if you measure glutamine levels in bloodstream, You can predict Alzheimer's disease 5 years before it shows its first symptom, right? So I'm sure that we don't have knowledge, but there are things which are happening. There are changes in our bloodstream, changes in something, and if we know exactly to what to look for, we can predict the disease long before it manifests in our body. We have some knowledge about few things, right? So, but cleaning it systemically, if it is not toxic to the body, is good. Microbiota is one of those things, eating prebiotics. So they don't have to go to the tumor site. They can just clean up the whole body, right? But there is no doubt about it that the microbiota at the tumor site is pro-tumorigenic. Go ahead.
There was a day you mentioned about oncolytic viruses.
Say that again.
There was a day you mentioned about oncolytic viruses.
Oncolytic viruses, yeah.
So how do they discriminate against the normal cells, and what is their effect? As in, what exactly do they do to the cancer cells, and what is their effect?
Yeah, so what are oncolytic viruses is that, huh?
In Kaposi sarcoma.
Yeah, so these viruses, I'm just trying to remember what is there inside, because I am thinking about oncolytic vaccine versus oncolytic viruses. So the oncolytic, so what happens is that the viruses which are there, so viruses are used as a vehicle to deliver the things to the tumor site to basically kill the tumors. Okay, so you know what is an oncolytic virus, so what is a vaccine, cancer vaccine, you know, right? So like many of you may have used lentivirus or retrovirus in your lab. I don't know if you have used, you have not used, right? So viruses are very useful as delivery vehicles. Okay, so oncolytic viruses are those viruses. So viruses, you have seen this kind of viral structure, right? So you package your material inside the virus, and virus can very easily go to the site, and if you decorate the virus with the right thing, it will go to the tumor site. Okay, and then it will release the cargo, and that cargo will kill the tumor cells. This is a very generic thing. Oncolytic virus will come in details when I will tell you about biologics tomorrow. So I will tell you exactly what it is, but this is a very simple version of it. And cancer vaccine, you know, the antigen against specific towards cancer. And so the immune response will be triggered, which will kill the tumor cells. So tomorrow I will show you a slide about biologics also. Biologics are like antibody-based therapy or cancer vaccine or oncolytic viruses or CAR-T therapy. All of these things will come tomorrow. So small molecules we know very well, right? Drugs. But these guys will come. There are 9 topics we'll discuss, fusion proteins, things like that. See, we are finishing up even the tutorial right now. Then we can really enjoy in the afternoon.
So you're going to give us all the slides after?
Oh, absolutely. You are going to get every single slide. This morning I woke up at 2. So I sent all the slides to Venkatesh already in advance. Yeah, so you will get everything, every single thing. I modified a lot of things this morning, and so yeah, you will. I have already sent him all the slides for tomorrow also. So he will send you everything. You had a question, yes, go ahead.
Mix is part of the signal transduction pathway. So, ma'am, it is a protein, then it will be only active when the signal molecule is binded to the receptor.
You're talking about MYC.
MYC.
Yeah.
MYC. So, ma'am, it was part of the signal transduction pathway.
So here is the thing. All the proteins are part of signaling things. Nobody exists in isolation. They're all part of some cascade. So, go ahead.
Generally, the proteins that are part of signal transduction pathway specifically, they are only in active form when a signal molecule is binded to that specific
So let me
pathway.
correct you here, right? Let me correct something here. All the proteins, none of the proteins are like one protein living in isolation. Every single thing is part of something. They are part of some signaling cascade. They can be top of the ladder, they can be the bottom of the ladder. So top of the ladder is receptor, which is starting the signal, and then it is going to the nucleus, and all of these things are in between, right? And nuclear proteins are there and things, and then the response comes in. So MYC is also part of a signaling cascade, right? But what happens is that MYC is a very unstable protein, okay? But, and so I was doing gene chip analysis when it was developed back in early 2000, and I could not detect MYC because MYC just degrades so quickly, right? It is a very unstable protein. And there was a big controversy going out at MYC levels, and we had developed this approach. We could have answered that. But even then, I could not even isolate MYC because of the unstable thing, right? But in cancer, MYC is really stabilized. It is really stabilized. So what happens is this, what goes wrong? So you guys really answered a lot of very good questions today. So think about DNA makes RNA, RNA makes protein, right? So sometimes the problem is at the DNA level, there is gene amplification. So every time you will make more RNA. Then sometimes the problem is at the RNA level, that there are microRNAs which will, even the RNA levels are same, they will degrade it, so it is no longer made, tumor suppressor is not there. Or they will increase the translation, more is this, or they will stabilize it, or they will destabilize it. So even if you make the RNA, you don't know how much protein you are going to make. Now let's come to the protein, and that brings me to MYC. So when MYC protein is made, it is degraded very quickly. Okay, but in cancer, MYC is not degraded because it binds to other proteins. And when it binds to other protein, it is extremely stable, like really, really stable. And then, so when you look at protein levels in the cell, this protein level, this steady state protein level that you're seeing, that is sum of synthesis and degradation, synthesis and degradation. But if you stop degradation, the levels will keep on increasing, right? Makes sense? So MYC is really stabilized in cancer, okay? And usually when it is in partner with its partner, I work with that protein, but right now it is not even coming in my head what is the binding partner. I think it is aurora kinase. So when it binds there, MYC is also highly active. Okay, so sometimes this is a very common theme in cells, that if there is huge excess of protein, they are very active also. They don't degrade, they are very, obviously they are very stable, but they also become highly active. Okay, and their conformational changes which keeps them in active state. So to make a long story short, MYC become independent of outside signal. When it is supposed to be degraded, it is not degraded. Okay, and like somebody told, who said about localization today, somebody said about localization today, right? So one of the biggest story in RAS signaling is that RAS is probably one oncogene which is expressed in all human cancer, and it alone is responsible for more than 30% of human cancers, correct? And RAS is a GTP binding protein, and GTP binding to RAS is so tight that nobody in the world could ever think that they could remove GTP from there, because it's picomolar binding, extremely tight, right? So when I started my lab at Purdue, so we were working with G proteins, which is RAS was one of them. So we developed a method there, and so I know a lot about the tight binding, and we could never do. But then the first time ever, one person actually developed an inhibitor which can kick GTP out of the pocket, and that person was my postdoc advisor's lab. Okay, so in his lab, what they did, they looked at the crystal structure of that, and so this is the active site, and this is where GTP binds. And remember, we were talking about allosteric sites yesterday. So he found that there is a small pocket allosteric. So he just made an inhibitor and accessed that pocket, and he could kick GTP out. And that was one of the biggest discoveries of our time. So, and he has opened a company, but since he has found, now people know there is allosteric site. So there are many, many inhibitors there, like many inhibitors. His first inhibitor is his, and he has a company, and I am sure it is going to clinical trials. Hi. So we have finished all the classes for today, and tutorial is going on. So yeah, really ahead of time. So everyone has afternoon off today. We are really huh? 4 lectures. One after the other, no break. Because there was nobody to tell us to take a break. So we went on. We rely on you to tell us it's break time. You were not there. We didn't know what to do. We knew lunch would not be ready. Let's use this time and then have a fun afternoon. Yeah, so coming back to that, now there are many inhibitors for that RAS protein. So, but before that, when we did not know, then we were not allowing it to go to the membrane, because if you don't allow it to go to the membrane, it is ineffective. So what you were saying, that it should not translocate there. If you can find translocation inhibitor, it will not be effective. So that is a very good strategy also, right? Absolutely, you can check for some product which is downstream. You absolutely can check, but you don't want to go too downstream, because it may get signal from many different ways. So you have to go downstream most of the time, 90% of the time. Okay, and what is a generic cancer marker that you can use for as a pharmacodynamic biomarker? Ki-67, that's very good. And what is a predictive biomarker?
Like when we predict the occurrence of, like, chances of occurring the disease much. There is one.
Don't worry about it. Take your time.
Okay. Prognosis and then predictive. So
Yeah, take your time. Take your time. Think about it. Let me guide you. I'm deliberately asking you in the wrong order. So let me ask you in the right order. Okay, so what is a diagnostic biomarker? Anyone? See, if you know the answer, you should grab to the chance. Then if you have given the answer, your job is done. I'm not going to bother you unnecessarily. Okay, go ahead.
Used to identify the presence or absence of the disease.
And is it treatment dependent? No, it has nothing to do with the treatment. It is before treatment.
which treatment to choose?
Yeah, no, you don't use, you can use it up, and of course you will identify the disease, but it is before treatment. What is a prognostic biomarker? Explain to me using an example.
Prognostic.
What is the meaning of prognosis? Yeah.
Curability.
Oh, you want to answer, right? Yeah, go ahead.
Prognosis is kind of a prediction about what the possible fate of the
Disease is. Yeah, it has nothing to do with the treatment. It is like, we are passing our expert comments on everything, right? It is like giving our prediction about the outcome of the disease, not about the therapy of pancreatic cancer, that reminds me, ChatGPT often gives you wrong predictions also. You give them and it seems pretty hopeless, and it is not that hopeless. So when you use AI to determine something, AI can be fooled very easily. So I have seen AI giving me different answers for the same questions, and then you add something more there, and now it will make the connection which is not even there. So it is so good in making all the connections which do not exist, okay, because it is just trying to find the connection. So be very cautious when you are using AI. It is very good in many respects, but it is not 100% right. Just like internet, not all the information on internet is correct. So yeah, you are right. You are right. Give me an example.
If it's triple negative breast cancer, we could give prognosis that it will be
Very poor, very poor. If it is P53 negative, it's going to be poor. If it is tumor suppressor negative, it's poor, right? And now with that, what is a predictive biomarker? Anyone?
Measures or it is used to selection of drugs.
Selection of drugs. You are predicting which drug will work better. That is predictive biomarker. You are absolutely correct about that. So, and what is a risk biomarker?
Before the presence of disease, we use it to identify if there is risk of that disease.
Exactly. So if somebody is BRCA1, BRCA2 negative, you say this person has a chance of developing, a risk of developing breast cancer, ovarian cancer, right? Yeah, that is a risk factor, right? That is very good. And then what was the other biomarker? Risk?
Safety.
Safety biomarker. Did we cover safety biomarker?
Yes.
So what is a safety biomarker? Even I can't remember that. I remember it was written in some figure, but I don't think I thought it was important enough to even, yeah. It is a kind of risk factor, I think. We can check it more. Yeah, it's a safety biomarker. It could be that, if this biomarker is present, maybe it is a good prognosis, something like that. We'll double check.
Then there was one therapeutic biomarker.
There is a therapeutic biomarker. What is therapeutic biomarker?
Like when we have given the drug and now we want to check is it actually binding the target.
Exactly, that is therapeutic biomarker, right. That is right. Now tell me 3 different ways health disparity works, or 4 ways. 4 things. You all can tell me different things. What leads to health disparity? A precision medicine disparity, not health disparity, precision medicine disparity. They are 2 different things.
What leads to disparity?
Precision medicine precision medicine disparity. So suppose an Asian person is there, an Indian person is there versus a Chinese person, right? So we have different genetic background, obviously, little bit of different genetic background, and so how is precision medicine disparity working there? Yeah.
In precision medicine, the sample size that we take might not be diverse enough for the treatment to be applicable to a wide spectrum.
Exactly, that is the first thing. So misrepresentation of minority groups, right? Usually they are not included there, right? So that is first. Give me the second one. Obviously money, not having access to precision medicine, right? Not having access to screening facilities, not having access to, not even knowing about things. I mean, how many of you have seen in your lifetime that you didn't even know about that opportunity? You could have availed of that opportunity, but nobody told you about that opportunity, right? We have all suffered in one way or the other, right? Because we did not even know the existence of that opportunity, right? You could have gotten somewhere else had you known at the right time, right? So if you don't even know about what exists out there, how would you even know to screen it, right?
Then benefit, whether they are able to take the benefit even if it accessible.
Which way? Give me an example.
For example, if the patient is not having PD-L1 expression, still we were giving him the immunotherapy.
Exactly. So how will the therapy exist, but they have not even screened for it. right? So that's where this story of this lady who had EGF HER2 positive cancer.
And
So they did not even bother to check. She had breast cancer. HER2 is a big reason for breast cancer. They didn't even bother to check because she was a poor lady, minority lady, and then they just gave her chemotherapy and she died. Right? And she would have easily survived with Herceptin. It is very effective. So, see, so many reasons for health disparity. And another reason,
Availability of the stuff at their residence.
exactly. And what else?
Food disorder.
Food, deserts, environmental, this thing. Like these, these Native Americans, they live in their own areas. So in US, they are given their own lands and nobody can go in their lands. So police and all these people are not allowed. That is their sanctuary. So, and they don't come out of those sanctuary, they live within their own tribe. Some people obviously come out. Once in a while you will see Native American among the common people, but it's extremely few people come out. So they live within their own cocoon, right? So they don't know much about it. And there are other communities in US also. They are called Amish. I don't know if you have heard. So Amish, they are very resistant to modern technology. So I have seen them from a distance. So there are there are few Amish communities near Philadelphia area. So they don't drive cars. They still come in horse cart because it is against their thing. They wear all these hoods in their head and they wear this long, something we see in movies, that those kind of clothes fully covered and all that's, the skirts are going all the way to the floor. Okay, and they don't speak to anyone, and they come sometimes in the main thing to buy something and they will disappear in their own community. So they don't have healthcare access. I don't know if they use any medicine. So these people don't have access, they don't have knowledge, they have real barrier to these kind of things. They think, I don't know what they think, but they resist any new technology, any new gadgets, anything. Forget about new gadgets. They are living like we used to, or they used to live in 1800 or 1900. So that's their culture. So these people don't have access. And then, when you live in a certain thing, it can lead to other kinds of stress, which can then lead to predisposition to other diseases. Have you heard of pre-diabetes? Many of you have heard of pre-diabetes, right? So now it is easy to diagnose diabetes before it develops into diabetes, right? So once you know that you are prone to, or you have pre-diabetes, if your sugar is 115, you know you are pre-diabetic. So you can take preemptive measures never to go to diabetes. So again, healthcare access, right? So you understand medical, this thing. Now, let me think of a case here. Suppose a patient comes to your clinic, right? And he is one of the minority person in the country, right? So let's say that person is of Chinese descent, okay? So obviously there are fewer Chinese here, right? And you are a doctor and he has some kind of cancer. So what will you do for that? So this person, let's say, have prostate cancer, or let's say he has he has this NSCLC, that the small lung, what is it that?
Non-small cell lung cancer.
Say that again.
Non-small cell lung cancer.
I never can say it right. Non-small cell lung cancer. He has that, right? What will you do as a doctor? Not as a doctor, as an oncologist. specialized in cancer center, what will you do? How will you go about it? He has lung cancer. You don't even know what kind of cancer he has. All you can see that the cancer is in the lungs. Go ahead.
Ma'am, as per the medical ethics, we can do two things. First, the autonomy, and we can go with the beneficence. In first case, we have to make aware of the patient, like what are the consequences, and if it is not treated, then what can lead his or her health. And in case of beneficence, we must ensure that the person whosoever is doing certain kind of surgeries must not be harmed, and also we have to make them aware of their further future consequences.
That is very good. But you made the decision for surgery, right? So before you even think about surgery, what things you have to consider, right? So anyone who comes in, we cannot do surgery on that person, because it is so. In prostate cancer, for example, when it starts, it is a very localized cancer, right? In breast cancer, initially it is a localized thing, so you can go in and surgically remove the localized thing. In lungs, it may be, at few different parts. So there are ways to, remove that, but it is a very aggressive way. So when a person, all you can see are spots in the lung. So you cannot really make a surgery decision right away. You are absolutely correct. When you are at that stage, you have to do exactly what you said. But what will you do the first time? The person is complaining they have pain in their chest, right? Just 1 second. They have pain in their chest, right? So you said, okay, skipping few steps, you have figured out it is cancer. Because how would you figure out it is cancer first? Like, what is the way to figure out it is cancer?
Biopsy.
You could use imaging tools to see if there are.
What imaging tools?
We could
I guess biopsy we do.
No. You cannot do lung biopsy without even knowing that it is lung cancer, right? What is the best way to conclusively say it is at least cancer?
And that cell differentiate nahi, but
PET scan. ma'am,
PET scan. But how do you do PET scan?
We would need to, if we are looking at the lung area, there will be specific fluorophores that would tag to the disease cells. And
No, you don't even know what is. See, to tag it to disease cells, you have to know the molecular target. You don't even know whether it is cancer at that stage. You are the doctor. You have to first figure out whether it is even cancer, right? And this patient is saying, I have problem in breathing, or what, I don't know how it manifests. But what will you do? Suppose it is breast cancer, we'll do mammography, right? And mammography detects lump, but lumps does not mean it is breast cancer, right? So how will you even know this is cancer?
Medical history.
No, medical history has nothing to do with it.
Any biomarker presence or absence of any biomarker?
Hey, you don't know, even if it is cancer, where will the biomarker come in? Right? First you have to diagnose it is cancer. Forget about breast cancer, lung cancer, head cancer. It is before you use the word C.
In PET scanning, they detect the glucose uptake.
They take the glucose uptake. That's the thing. And you use radioactive glucose, and cancer cells love glucose, love glucose, love glucose. That area will light up. And that is a generic thing. All cancer cells will eat glucose, correct? So you treat the patient, and you really hope nothing shows up. You really, really, really hope to tell the good news to the patient. They don't have anything. We don't even use the C word when actually the person has it, right? So you are good and to go home and enjoy life. But if suppose something shows up, that tells you first it is cancer, right? So now you have diagnosed that it is cancer. What is the next step you will do? It is showing in lungs. Anyone?
Prognostic
To know at which part of the lung it is affecting.
So that PET scan will show, right? That PET scan will show. What is the next step there?
we need to find the
You-
extent of how.
That it will show. That it will show, but what will you do next? I am showing you the PET imaging. Okay.
Check TNM. That was tumor node and metastasis. The status.
Absolutely, very good, very very good.
We'll
So
check the status of which
that is awesome. That is awesome. You will see whether it is localized, whether it has spread, whether it has spread to lymph nodes, whether it is metastasized to other organs. Great answer, right? So we will try to determine how intensive it is, right? Is it a one spot in the lung, or is it spread out, or is it has reached lymph nodes? Lymph nodes, even if you don't know what it is, I'm going to show you the picture in one of the lectures. So lymph nodes are part of the lymphatic system, and on an average we have 500 to 700 lymph nodes. They are just like small centers, and they are all joined together by the lymphatic system. So when cancer spreads, it can go to lymph nodes. It often goes to lymph nodes, which is a very bad prognosis. So yes, you will see, you will do the TNM staging of that. Great. What next? Give me exactly the answer, the thing. What things? So have you used diagnostic biomarker? Okay, you know the answer, right? So think as a doctor, think as an oncologist, right? So you did the PET scan, correct? And you saw the thing, and by the PET scan you know whether it is in the lymph nodes, whether it is not in the lymph nodes, right? So you have all that information. What is the next thing you will do? Any of you? You guys are the oncologist here. So how will you best treat the patient?
look for a size maybe, and primarily look for.
So I should probably ask you guys to tell me your names when you answer it, because I am very poor with names. So I have to hear 3-4 times before it actually registers to me. Because I am so tempted to ask your name, but then I don't ask because I know I will not remember. But after a while I'll remember, so I don't want to disturb you guys, but maybe I'll ask you separately. Yeah, please answer.
My name is Shivani, and my answer could be we would look for the angiogenesis status, or maybe the size of tumor, if it's curable, if it's surgically removable or not.
Yeah, that is very good. If it is localized, then that is very good. That's when surgery will come. If you see it is only in one place, it has not metastasized, surgery is such a wonderful option, right? That's when you will kick in. Everything that you said will come into picture. That is actually a very good prognosis. It is a localized disease, right? And so that you will do surgically remove it. Okay. And suppose we do surgery and we remove it completely, that is great. But there's something else you might also want to do before you go for surgery.
Yes, we will eradicate with radiations before doing surgery, and when we have isolated that tumor, we'll do eradication again just
to make sure that, and sometimes we also give chemotherapy just in case something is lingering. Great job, guys. We will do that. Right? That is very good, and we will make sure that the patient is aware of all the things. And it is also the doctor's job to sometimes drill on the patients that how bad it can get, because they don't understand. So you have to be very honest in giving them your honest opinion about that. And sometimes it is even bad to wait for 1 week in such cases. You have to treat it ASAP. Yeah, so we have to do everything. We have to tell them the pros and cons. We have to tell them the side effects, potential side effects and everything. But we have to also cheer them up that they are up to that thing and it can be cured. And not be negative, even if it is negative prognosis. Yes?
Can liquid biopsy replace PET scanning?
In some cases it can. But PET scan is very essential to see the extent of metastasis. So PET scan I would not avoid. What else? You guys are forgetting something very important there.
Expression of tumor suppressor gene.
So suppose it is P53 negative, would you directly go for P53 negative thing? So suppose it is lung cancer, right? What is the you can see that it is in the lung. What will you do next?
EGFR testing.
Yes, correct. So there are different kinds of lung cancer, and so they all have their own markers. So this non-small cell, whatever, NSLC, this one they have its own markers. So then you will first test the markers there, exactly what kind of cancer it is. This is very important. If this is a breast cancer, there are so many kinds of breast cancer: HER2 positive, TNBC, luminal breast cancer, basal carcinoma breast cancer. There are just too many things. So the very first thing you will do is to find out exactly what kind of breast cancer it is, what kind of lung cancer, what kind of liver cancer, what kind of brain cancer. So that comes really, the moment you do PET scan and you confirm it is cancer, the next thing is to find out exactly which cancer it is. Before even you make the decision to do surgery or chemotherapy or anything, the very first thing is that what is your enemy? You have to recognize the enemy before you start planning targeting the enemy. So you have to find it, right? And if it is in lung, and in lung EGFR is mutated or deleted, that 19 exon deletion, you are going to check all these things. So that will actually tell you if it is, what kind of lung cancer is that. So we have to do that ASAP. We don't want to go for chemotherapy or even surgery, because surgery is also very bad in some cases. So why it is bad? Even if it is localized cancer, right? When you are surgically removing it, some cancer cells may escape and go in the bloodstream, and then the whole body will have metastasis. So surgical thing is also hard. You don't remove the tumor, you also remove part of the organ, just to make sure that you contain the tumor inside the organ. So surgery is a very big step. So you don't want to go there without knowing exactly what you are dealing with. So you have to really identify the kind of cancer. And then you plan your surgery or radiation or chemo or that. And then once you know which tumor it is, what kind of it, you look at, the real markers. Tell me the real markers. Once you have said, okay, this is this kind of cancer, then what are the other things you will look for it? Which is common across cancer. No, not a mutant. So what is a common thing cancers do to survive?
Suppression of
Immune system. So you are going to look at, do they have PD-L1, right? Because that is something they commonly use, right? So PD-L1, and then there is the CTL4. I will tell you in details once again about CTL4. So you will look at the status of these things, right? So then, if suppose PD-L1 is positive, then you can immediately give immunotherapy to that person, right? Because even if something can be surgically removed, if you can shrink it, it would be easier to remove without damaging the organ or without removing too much of the organ. Do you understand that? So you are going to look at all these markers, which is common marker across the cancer, right? If it is in the lung and if KRAS is mutated, you will look at the KRAS status there. And that will tell you how aggressive or non-aggressive. That is when we will look at P53 status. If it is breast cancer or prostate cancer, we will look at BRCA1-2 status, right? So first you have to know your enemy properly before you can deal with it. And then you design your therapy. And then you give the drug, but you monitor the patient very closely. Is it regressing? Sometimes you can see visibly that it is regressing, but if it is deep inside the body, then you have to keep on regularly doing PET scan to see if it is shrinking, if it is not metastasizing, if it is stable, right? You guys are oncologists now. The patient comes and you know exactly what to do, right? Yes.
Ma'am, in what cases do surgeons usually avoid not surgery?
Say
Not
that
doing
again.
surgery? In what circumstances does surgeon resist doing surgery?
That you don't want to do surgery?
Yeah, like they say, no, we can't treat it, we can't put it out now.
If it has metastasized, no surgery.
Right.
Because in metastasis, surgery will not help. Because in so many parts of the body, what are the things you can do? So in metastasis, they won't go for surgery.
Okay.
So they will just give systemic therapies, immunotherapy, angiogenesis inhibitor therapy, these kind of things, chemotherapy, but they will not do surgery.
By any chance, like, is there risk of the cancer cells seeping into the blood vessels again while doing surgery?
Oh, definitely.
Right?
Definitely there is a chance. That is why they remove part of the organ also,
Right.
so that, they don't, there is no chance of doing that.
Spread of
That is why they also treat, this folate receptor therapy. So cancer cells overexpress folate receptors. So you make folate receptors bind folate. And folate is a small molecule, right? It is a common thing in our body. So in under normal circumstances, folate receptors are present in very small amount. So if you make folate attached to a fluorophore like FITC or rhodamine or something, and then you can use it during surgery. I know it is approved in Europe. I don't know if it is approved worldwide. It is not approved in US as far as I know. So you can feed the person there, and then the cancers will light up, right? And then you do surgery. So because that will enhance the sensitivity tremendously, so you can do a better, more precise surgery. And I think now that these robots are involved in surgery or something, I'm not exactly sure. That's not my expertise, but surgery comes with its own risk. So you don't want to go to surgery, but if surgery can remove the cancer in one shot, why not? Why not? So people do radiation and surgery all.
They are also developing some kind of nanobots.
Huh.
They
Nanorobotics.
implant some sort of nanobots to the side, and these nanobots are performing the whole thing.
For example, in that vessel blockage, the blood vessel.
Yeah, they do that all the time. Yes, you're right. You're right.
Ma'am, that universal biomarkers, I guess I missed that thing.
Universal biomarkers are like PD-L1 biomarker, right? PD-L1 is overexpressed in many tumors, so it's considered as universal. The chances are it will be across the diverse thing.
The Ki-67.
Ki-67, you are right. It is a universal biomarker, pharmacodynamic biomarker.
Actually, what is this Ki-67?
Oh, Ki-67. Oh, what is Ki-67? So you tell me what kind of biomarker would you use for cancer cells? So if you have, let me walk you through. If you are telling to a kid what is cancer, what are the things you will tell him in one sentence? What is the first thing you will tell about? I hope you don't have to explain it to a kid, this thing, but if you have to tell somebody who is a layman, right, and you tell what is cancer, how will you explain it to that person?
We will specify the difference, what is normal cell and what is...
No, tell me in one sentence, what is cancer? Uncontrolled cell proliferation, right? That's where the cells are dividing. That's the definition. You don't have to say anything beyond that, right? And it forms a tumor. That's all it is. So when cells are dividing, what is happening all the time, which is not happening in normal cells? Forget about the details of EGFR and BCR-ABL and Gleevec. Forget about everything. Cell proliferation, what happens when cells divide?
Ma'am, CDK2 kinase वगैरह सब they get disturbed, then they get...
Cell cycle kinases are hyperactivated. And what are the process of cell division? So if you look at cell cycle, go back, cell cycle. What happens during G1? it's again a rest period where they're getting ready for mitosis, and mitosis is separation, the everything, all those things, the mitotic spindle formation, proper separation, and then anaphase, the cells actually completely divide, right? So out of this whole thing, DNA replication during S phase, so the cells which are dividing very rapidly, DNA replication is occurring all the time. So Ki67 protein is part of DNA replication machinery, and that is why people look for Ki67, because Ki67 levels are really elevated in cancer cells because they are dividing all the time. And then something you may not know, even I did not know. So one time I acted as a consultant for a company who was working on aurora kinase. So that's when I came to know about it, because I was reading about a lot of things about aurora A clinical trials. And that's when I came to know that these cells that you guys grow in the lab, so usually they divide every 24 hours. You know that, right? Yeah. Roughly 24 hours. But human cells, even if they are cancerous, they are not dividing every 24 hours. They take much longer to divide in human body. So some cells divide, cancer cells divide obviously at a rapid rate, but normal dividing cells in human body, in a normal human body, sometimes they divide once in 4 days, or it takes 4 days to complete the cell thing, or sometimes 5 days, sometimes even 10 days. Okay, so they are not dividing every 24 hours. So when you use anti-mitotic drugs, they work in cell culture much better than they will work in a human body, because cells are not that rapidly dividing. Okay, so basically Ki67 levels are much lower in normal cells compared to cancer cells. So that is why Ki67 is a reliable marker. Okay, to see whether cancer cells growth is inhibited. Go ahead.
Coming from when the cancer tumorigenesis begins, is it the right way I'm thinking? The more size grow, they get more stronger connection with the rest of the tissue, with the matrix, or does it weakens out? Or just to metastasize to the different location, they have to break themselves at the site of their generated to metastasize to somewhere else. So is it right that as their size is growing, they are making more stronger connections with the rest of the surrounding tissues, or they loosen it?
So what happens is that when the tumor forms, right? So initially they are together, right? They are together and they are loosely held. They are not very tightly held. They are loosely held because their differentiated cells are properly arranged like bricks. But here, if you are putting bricks on top of it, you can make a wall very haphazard. Cancer is like that. Nothing is differentiated. It won't look like brick. It will look like something is this, something is this. You just somehow this thing. So that is very leaky kind of thing. I will tell you about an EPR effect later on. So then you will see cancer cells are very disorganized. They don't have a nice clean structure. Okay, so they are not extremely tightly bound like normal epithelial cells. Having then on top of that, if this is the tumor, these which are on the outside, they change morphology and they become more mesenchymal so that they can escape that and metastasize. So in general words, they tend to be more loose on the outside so that they can go and form a new colony elsewhere. I mean, this cancer is, it teaches us so much more. It teaches us, I mean, so many different kind of things. Okay, it is amazing, but it is just really bad because it is such a devastating disease, but it has told us about so many new things. So one of the things is that, during embryogenesis, we have stem cells, and that is how we grow all the organs, right? And then what happens is that as we are born, slowly stem cells recede. And so in an adult human body, we have very few stem cells in different niches in the body, and they are there to protect us from, if we have damage, they will come and repair it, right? So that's good. But we have very few stem cells, and stem cells are really very powerful. So those stem cell pathways are shut off in human adult body. Adult means after birth, right? And cancer cells will reactivate those pathways, the embryonic pathways. So at embryonic stage it was great, but in adult when they make it, that helps cancer survive, and that is bad. Makes sense? So for example, I'll give you a different example also. So CDK5 kinase is super active in embryonic stage, okay? CDK5 kinase is the one which builds the whole brain, and brain has extremely complex structure, layers, this layer, this layer, this layer, this layer, like that. So if you delete CDK5, brain doesn't even form. So very high CDK5 activity is required. But upon birth, CDK5 activity declines, and then it is a steady state level. If it is less, then that person will have neurodevelopmental diseases like schizophrenia and ADHD and autism kind of thing. But if it is too high, it will degenerate the brain. But here is the thing, that same high CDK5 activity in embryos was beneficial in making the brain, but in adult, that high activity degenerates the brain. So what is good for the embryo is bad for the adult body. So my friend Dr. Hisanaga from Japan, he was the one who actually showed that why high CDK5 activity in the embryo is not bad, and why it is bad in adult brains. This is a other mechanism. So what cancer cells do, they turn on a lot of embryonic pathways which shut down after the baby is born, and then they exploit it to develop cancer, and that is very bad. So they hijack a lot of pathways. So cancer cells have taught us so much about human things. But you know, it is a very bad disease. But the good thing is that it is a preventable disease. It is really a preventable disease. And that's the beauty of it. There are 1,000 players involved, or maybe 2,000 players involved in developing a cancer or preventing a cancer, but we don't have to worry about who these 1,000 players are. We just have to live the life in the right way, and our body will take care of everyone. If you go to Europe, people still smoke a lot there, right? Young girls, young kids, in bus stops, everywhere they are smoking, train, everywhere they are smoking. So when you are exposed to this kind of thing, you should not stay there. It's really bad for you. You have to avoid smoking. You know, I don't know now, when I was a kid, there were places in trains where it used to be written Dhoompaan Nishedh, right? So it used to be written there. So I remember I was a kid. Obviously, I didn't know anything except that cigarette was very bad. So I was a kid, and somebody sat there and in a train, and he started smoking. And I said, "Aapko dikhai nahi deta? Ye likha hua hai." And I was 9 years old. So मतलब he was so shocked कि बच्चा उसके पीछे लग गया है, you know. But I'm just saying that, you know, take your stand. Don't expose yourself to that. You know, so I'm in Cancer Center, and they organize this yearly meeting in Cancer Center where they bring some extremely famous people who are cancer survivors, like extremely famous people, like Olympic gold medalist kind of people, right? So one time they invited some lady, and I don't know who she was, but she had achieved something really great, like Olympic gold medalist, but I honestly can't remember, but she was a breast cancer survivor. So she was telling her story that as a young girl, she was working in a bar to make some money to go to school. So she was bar smoking is allowed. So she was exposed to this second-hand smoke, and then she developed cancer. And then she said, I was not even involved in smoking. I had nothing to do with that. I was a poor kid who was trying to save money to go to college, and I got cancer in return. And then she achieved something great in life. So smoking is pretty bad, and there are people who do smoking plus alcohol. It is a point of no return. Once cancer is there, cure is also not an option many times. Go ahead.
From the lecture, you told us that the lung cancer is the one that is topping the list.
Yes, in terms of number of cases.
And there's a lot of energy, a lot of research, a lot of money is put in.
Exactly, yeah.
Yet we can just avoid the business of
We can avoid the whole thing. We can avoid the whole thing. You are absolutely correct. You know, the more I study about all of these players, like infinite number of players involved in cancer development or Alzheimer's and everything, and what I think is that we exercise and we don't have to worry about what is changing in our body. We are just exercising, and all the right things come into play because the body knows which to inhibit, which to activate. We don't have to even worry about it. We can watch a TV show which we love while we are exercising. We are being entertained, or we listen to the music that we love. You know, anything. I was telling my friends here at IIT Indore that I hate exercising. I really hate exercising, but I exercise every day, and now my reason to get up in the morning is exercise. People think of tea, and my motivation, oh, I will exercise in the morning. And how I came along this way, COVID played a very good important role, because I was bored to death at home, so I thought I better exercise, and that's how I started it. But I have linked to something I like, so I rarely watch television because, then I feel bad about it. But I like to see mystery things, right? So mystery things are good in a way, so I have linked to guilt-free TV when I exercise. So the more I exercise, after a while your body will crave for exercise. So I exercise for 45 minutes, and then I link it to something I can watch without feeling guilt about it. So now that keeps me motivated, because next day I will think, oh, so what happened next? Because it's a mystery thing, right? So I'm always, but I will never see it, no matter how much it is, because without exercise, I'm not allowed to see it. That I have put it on myself. So that helps me motivate. So if you don't like exercise, just link it to something that you love. I don't like to eat good apples, right? So when I go to work, I only take apples. Now, if I'm hungry, I'm forced to eat apples there. At home, I have a choice of picking things, but there I'm hungry, I have to eat apples, right? So you have to trick yourself to do that. And now I love apples, by the way, because, after 2-3 years of that kind of thing, I've developed a taste for apples. So we have to somehow coax our body sometimes, coax ourselves to do the right thing, When we work very hard and we have deadlines, then I say, oh, what after the deadline? I'm going to do this. And then I'm motivated to even work harder to go through that thing, right? So we have to we are human beings. We have to be rewarded in some way, right, at the end of the road. So you can develop your own motivational things, And at the end of the day, when you exercise, you feel so great about stuff because your brain is healthy and everything. And take my word for it, you will crave exercise, I started with 30 minutes. 30 minutes was killing me. And then I started elevating it. because if I don't elevate it, I don't feel like I've exercised, you know. So it is just extremely intoxicating to exercise also. It just makes you feel, it makes you feel you are tired, but you know, it is also exhilarating at the same time. It may take you 6 months to get there, but once you get there, that becomes the reason to do that. You can just, I'm sure you love music, you hear music, whatever. For me, it's, you know, mystery things. So,
But like people who tend to go to gym a lot, they have a high risk of heart disease.
but they also prevent heart disease. They also, because you know, I went to my doctor, right? So, like I was not able to climb stairs before since I was a young child, right? I could not climb stairs. So she told me, I was telling her my routine, and she said, you probably have a very strong cardiovascular thing. The funny thing was that when I went for my checkup, that was annual checkup, so I had a problem that I was feeling very tired all the time. So if I sit on a sofa, it would take me an effort to get out of the sofa. So I said, I don't understand. Like, my house is 2-story house, right? So I go up and down all day long. But it was becoming a chore to go up. So I would prefer to stay downstairs. But you cannot stay downstairs all the time. You have to keep going up and down. So I was feeling that I cannot climb 4 stairs, but I can exercise there every single day. So I said, why this is happening? And she said, so I said, this is very weird that I cannot. My thyroid medication was off. Like, you know, your body is too lethargic to do that. So, and she changed it and I'm back to normal, right? So I was able to do exercise because I love it too much, right? But I was not able to go up and down the stairs. So coming back to your cardiovascular things, you obviously have to work according to your body. Like suppose I don't exercise for very long, for what, like I have come here, I'm not exercising, right? So I go there, I may not be able to start exactly at the same rate. So I may be a little slow in the beginning. And then after a week, I will come back to my usual self, right? So you have to always look at your body and not push too hard. There are times when, you know, I realize it is hurting me and I know my body is disciplined enough. It is always telling me the right thing. I'm not making excuses for it. So if you don't feel something is right, you pause, you make sure. So don't go for that, I have to make the muscle and against your will you are doing that. Don't do that. And if you don't know, you should probably take advice. You know, unless you know your body well, you should take some advice. But there is no doubt about it that if you do exercise, you decrease the risk of cardiovascular diseases. But you know, I know of people who have fallen dead in the gym. There could be a lot of other factors, but exercise will not kill you. You know?
Yes, ma'am, I have also seen some. Ma'am, I am into the professional bodybuilding from past 3 to 4 years, and I have seen so many people who were having high BP, high cholesterol at the age of 40 or 35. But after doing continuously from 2 or 3 years gymming, now they are free from high BP, cholesterol, and they are living absolutely healthy lifestyle.
See? I mean, this is really amazing. This is really amazing that such easy things can fix everything in our body. You know, I work with diseases day in and day out. And what I have come to learn about it is that, you know, my diet, I never ate, you know, junk food anyway, but just improving the diet has such a tremendous impact. I have more energy than the people who are, you know, a lot younger than me because I'm very cautious about what I eat. Or I have more energy than the people in my age, you know, because I know what goes inside my body, right? I really make sure I never or I extremely rarely eat out. Like when I'm eating here, it's after years and years. Only when I am forced to eat outside. I never eat out just because I have no control of what are the things they are adding there. More oil, preservatives, everything is bad for me. So if you guys, I mean, I know you guys don't have a choice, and I shouldn't say that because you guys are forced to eat in the hostel, so in the mess. So, but the thing is that in the mess, I'm hoping they are not putting anything bad for you. It may not be tasty, but it is not bad for you. I'm hoping they are not putting preservatives there because they're cooking fresh food for you, right? So it may taste bad, but it is not unhealthy food for you, right? I know it doesn't taste good, right? But that I have also eaten here. This is my third time here, so I know pretty well about this place. Okay, but and I was in one of the faculty apartments last time I was here in 2019, so I am aware of your mess food, okay, but it is famously known, right? So, but it is not unhealthy for you, but it is up to you to make sure you are still getting enough fruits there. But this is very good to hear. This is really good to hear.
And also in India, the most of patient, as you have told, the cancer cells love sugar. So the Indian diet is every second diet is based on carbs, not on protein. Like
oh, absolutely,
yeah,
absolutely.
that's why the Middle East people or the African people suffer less as compared to the Asian people.
That is true. That is true. Like here, when you get tea, you don't even have a choice to add separate sugar because sugar is already added here, right? So how much sugar we are consuming, and that is very bad for us. Like the kind of tea sugar we are taking, and for a person like me who loves tea, I cannot afford to have sugar in tea. I really can't, you know. So yeah, you are right. We add too much sugar is one of the reason that, you know, for us, for Indian people, tea is one of the culprit because we add too much sugar in the tea. Tea is not the culprit, sugar in the tea is the culprit. So you're right. And then we tend to love more oily food because it tastes good, right? But it is very bad. But you know, nowadays you don't have to use oil and you can make equally tasty food. People are realizing that. You can make everything with less oil. I can tell you off the record, I can teach you how to cook food without oil. Seriously, I can teach you, and it will. I will tell you it is better than when you deep fry in oil. Any other questions, guys? Today they have really forgotten us. Go ahead, go ahead. Just give me some, I'll get some water. For you, I have to come close. You are the most soft-spoken person here.
Talking about sugar, we know that in the body, the sugar that is used is glucose.
Yeah.
While the sugar that we take is in the form of, say, sucrose.
It is sucrose.
Starch also. Then there are so many other sugars that are taken. So when you're telling people that don't take sugar, and the specific sugar that is used in the body is glucose.
Hey, so what happens to sucrose? You are a chemist, tell me. How is sucrose decreased?
Sorry?
Sucrose is a mixture of what sugars?
It's a mixture of fructose and I think glucose.
Yes. So you are creating?
Yeah, so even starch it also later converted to glucose.
Yeah, and then, okay, tell me about glycolysis. What happens in glycolysis? So if you start with glucose, what happens to glucose? Glucose is converted to glucose 6-phosphate. What happens next? Glucose 6-phosphate is converted to fructose 6-phosphate. So if you start with fructose and glucose, basically it's the same glycolytic cycle, right? So sucrose is the same. If you drink milk, milk has which sugar? Lactose. And when lactose is decomposed or degraded, what are the sugars? Huh? And galactose is isomerized to glucose.
So when you say that
Galactose becomes galactose 1-phosphate.
It's a main sugar, right?
No, lactose is the main sugar in milk, and lactose, when it is degraded, it is glucose plus galactose. Galactose becomes galactose 1-phosphate, and galactose 1-phosphate is converted to glucose 1-phosphate, which isomerizes to glucose 6-phosphate. It comes to glycolysis. So, answers your question? So, sugar is sugar. Whether it is sucrose, whether it is fructose, whether it is glucose, it feeds up into the same thing, right? Sugar is truly bad. It's truly bad. Nothing is bad in nothing, huh?
because sucrose is converted to fructose and glucose, you can also say let's avoid milk.
No, so that is what I'm. No, I was going to say this next, right? So anything in moderation is not bad. Even oil in moderation is not bad. If you don't take any oil, your skin cells will, this thing, you need some oil for skin, right? You know, they did I read somewhere, so these flaxseed, you know, flaxseed oils, capsules you get, right? So somebody was feeding their animals flaxseed oil. So I'm a vegetarian. So they tell you you should say fish oil capsule and flaxseed oil capsules. So fish oil is obviously better than flaxseed, but I prefer to take flaxseed just because of the vegetarian thing. So yeah, omega-3. Right, for that thing. So somebody had written that they fed flaxseed oil capsules to the dogs, to their pet dogs, and the skin became so good, really, really good, and all the warts and everything disappeared, right? So everything in moderation is good. Lactose and milk has so many good properties with calcium and everything, so you should definitely take milk. And milk is not a source of sugar. Lactose just happens to be there. Milk has so many good properties there. And most of the things in the raw form is good. Instead of eating protein powder, eat dal in the raw form. That is good. Because when you process it and process it and process it, you don't know what is going on, what is changing. It is also changing the bioavailability of the things, right? So things you take in the raw form, that is hardly ever bad for you. We take fruits, right? Fruits are full of fructose, but that's not usually bad for us. Unless somebody has severe diabetes, then they also limit some fruits for you because you can't even avoid that much sugar there. But they don't ever say that don't take milk. Milk also has fat, right? So you can say, well, you are eating fat. You have a choice between whole milk and, you know, fat-free milk or low 2% milk. I prefer proper milk. So it's your choice, whatever they like. So again, in moderation, nothing is bad. You should eat ice cream also, but in moderation. In moderation, everything is okay. We need carbs also. Yeah, we need carbs also, but we need everything in proper this thing. As long as you have discipline, you don't have to count your calories ever, you know. And if you want to indulge, eat the sweet also, but again. Yeah, go ahead.
Ma'am, like in post-pathogen interaction, so
What?
in post-pathogen interaction,
Yeah.
so there is a theory of co-evolve, like when pathogen gets upper hand, the host evolve. When a host gets upper hand, then the pathogen evolve. So in cancer case, so cancer does like in exosomes, so they have some microRNAs which changes like macrophage M1 state to M2 state, which is the anti-inflammatory.
Exactly.
So is there any is there anything like also our like human body evolve in such a way that it can treat cancer when it is present?
Oh, it definitely does. So, you know, like every second in our body, our immune system is cleaning off cancerous cells. So, as we are sitting here, a lot of cells are becoming cancerous. and they are being eliminated every single day of our life. Okay, it is when the immune system is exhausted, that's when it can develop. So this is a lifelong process, even as a young child, because oxidative stress is being created, genomic instability is happening. Who asked me why leukemia occurs in younger people only? You asked me, right? So I checked yesterday because I had no idea. I knew that leukemia occurs in younger people. And so what it told me, that leukemia occurs in younger people because hematopoietic cells are dividing very rapidly at young age. Everything is very rapid at young age because they're growing, because they're replicating so fast in a young age that they make replication errors, and that accumulates and that gives rise to leukemia. Okay, so body is constantly fighting. Anytime DNA error occurs, there are so many correction mechanisms. So basically we are eliminating cancer cells every second in our body. So body is always fighting, and when we are giving cancer therapy like immunotherapy, we are not doing anything. We are just using our own body against the cancer cells, right? We are just waking the immune system that fight it off. So body has enormous capacity to fight it off. So it's your body fighting for it. We are just giving it. So that is why the body has to be healthy, and if there is too much stress and the body itself is going down, you have seen that all of us know that when we are stressed, we start getting cold or something, our immune system is down, it is exhausted. So yeah, it is fighting, don't worry about that. We don't even know how many ways it fights. Yes.
So we know that all organs, they have their own cells specific to that particular.
Exactly.
So when a cell mutates and becomes cancer, so does it retain its originality? Such that when it goes to another location that is not that organ, you will be able to know that this is from a particular organ.
Oh,
say lung cancer.
Yeah, that is a very good question.
Yes, can we say that if conditions permit, a cell of a different organ can grow in a different place if conditions permit, if the cell is retaining its originality?
That is a really fantastic question. I would think so. So for example, when breast cancer metastasizes to the brain, to the bones, they also metastases to the brain, by the way. Breast cancer goes definitely to the brain. That is their yet another favorite place to go, to the brain. So breast cancer cells metastasize. You can see it on the bones very clearly that these are breast cancer cells. They have weird morphology and they are not differentiated as much. Today I showed you one mechanism for, what was it, where I showed you 3 examples. IDH1 makes 2-hydroxyglutarate. So one of the things 2-hydroxyglutarate does is to block differentiation. So cancer cells can become a little bit differentiated, but if they becomes differentiated, they are no longer that aggressive. So cancer cells tend not to become differentiated. It's because if they are not differentiated, not breast-like, or not basically keeping the original, parental phenotype, the more aggressive they are. So, but they still retain something of their parents. So you can easily recognize those guys outside of their original thing. But they are still cancerous, so they are not actually breast cancer cells, real differentiated breast cancer cells. But they are there, and they never form a nice layer. They are on top. They are just a lump of stuff. So they are good for nothing, because they don't do any good job. They are just there to steal nutrients, to invade, to just fool other cells around them by making them part of their own culture. So even if they are good, they were originally good cells doing their part in the whole physiology, they are not doing anything there. But they do retain some characteristic that you can say that they originated from where. Yeah. That's very nice question. We are supposed to have samosa right now. Right? This is very funny. Actually, we are left alone today. I have to say I'm enjoying it very much today. It is there?
Yeah, he's standing outside.
They are probably arranging our tea. Go ahead, next question please.
Ma'am, like in research purposes, how authentic it is to use cell line instead of primary cells, irrespective of cancer?
So repeat your question. How authentic it is used to cell lines? I missed the last part.
Use cell line instead of primary cells.
Oh, primary cells are very hard to use. It is very hard to use. It is very expensive to use. So you should definitely use cell lines. You should do most of your experiments in cell lines, because for sanity's sake, primary cells will drive you crazy. But once you figure out the whole pathway, everything, then you use primary cells to do the key experiments to confirm. You can get breast cancer or whatever cancer you're working on, you can get the biopsy tissues to figure out is it true in the actual patient samples. So you go there to do the final testing, then you go to the animal model. You can use patient-derived tumor cells to study in animal models, but you should, in my opinion, start with the immortalized cell to figure out the signaling pathways or the details. If you start with those things, first of all, nobody can afford, very few people can afford, and the difficulty level is extremely high. Sometimes even cells drive you crazy, so imagine working with those cells which will drive you crazy. You cannot even do the experiment, you just have to keep them happy all the time. You know? So cell line itself is a very good tool and you should make best use of it. It depends on the cell line, and also it is advisable to use more than one cell line to confirm it, that what you're getting is not specific to that cell line. So suppose you are working on triple negative breast cancer, you have to use triple negative breast cancer cell line. If you are working on castration resistant prostate cancer, you have to use castration resistant prostate cancer. And because no cell line is perfect, you have to use 2 or 3 different kind of castration prostate resistant cancer cell lines. And then in one P53 is there, P53 is not there, so you compare what's the difference there. So cell lines are so easy to use that you should just really milk it out and then go to finer models. So then your research will be very detailed and really validated research. And then obviously you have to go higher into actual patient-derived cell lines and things like that. Yeah.
Why do we go for adhesive cell lines?
Which cell line?
Adhesive, like we
Oh, adhesive cell line. It just depends on, adhesive cells are so much easier to work with than suspension cell lines. But if you are working with, Jurkat cells, right, for T cell signaling, you have to use Jurkat cells. We work with NEPC cell line, and NEPC cell line, the most famous one is HL60 or something like that. And you buy it from ATCC, it's around $1,000. And we have bought it 3-4 times, we cannot grow it. They are not adhesive. We just lose them after a while. Then we buy them again and we just lose them. And it is just very hard. Jurkat cells are not that hard to grow, but these HLL60 cells are very, HLL60, or I can't remember, whatever it is. That is the most famous NEPC cell line. And it is not my lab. Everybody has problem growing those cells. So it is our good luck that they are adhesive because they are so easy to grow. Some cell lines are okay in suspension, but adhesive cell lines makes it so easy. It's just so easy to work with
But they're
them.
very easy to die also. I mean, they have
No. They are not easy to die. You are doing something wrong there. Trust me, they don't die.
Ma'am,
You're,
they
if
must be right. You must be right. I must be doing something wrong.
yeah, you are, because because in your case it is good news that you are doing something wrong, because you can obviously fix it,
Yeah.
right? If it was an inherent property of the cell line, then you are in trouble, because then you cannot fix it. The good news is that they should not die. They don't die. You have to be really bad to them to kill them.
What happened?
What I'm saying is that don't take it otherwise. These cells are very resilient, they don't die. Never let them go over 50-60% confluency, never ever.
Okay.
Yeah, see, I tell my students, if you have crossed 60-70% confluency, throw the flask.
It was 100% confluency.
Yeah, huh?
It was almost 100%.
Oh, they will never survive.
Yeah,
And if you grow to 100%, those cells are nothing but a piece of protein factory,
yeah, absolutely.
because all their signaling is broken forever. You cannot retain those cell lines. So you never ever let them cross 70% confluency,
Okay.
and they will never die. And I would not, I mean, if you are doing signaling, your cells should never cross 60% confluency.
Okay.
Yeah, 100% is unimaginable thing. They are piece of garbage at that time.
Right, I get it.
And then you take it from 100% and you make it 20%, 30%. No, once they go to 100%, they are tainted forever. Just don't even try to split them. So,
Okay.
yeah.
Because we forgot to split them initially. By then, like we missed one day.
Yeah, it
One
happens.
day it happens.
You are not the only one. I'm sure it happens with all of us. All of us have made mistakes. But it is just better to know never to make that mistake. If it happens, it happens. Okay? You always have more vials. But don't try to rescue something which has gone 90, 100%. They are beyond rescue. And you are sowing trouble there. Because if you rescue those, the experiments are not going to be trustworthy.
One more question.
Yeah.
We borrowed the cell line from JNU.
Yeah.
And by some malfunctioning, our incubator got disturbed. So
Which happens all the time.
The temperature went for 20 degrees Celsius for the whole night, and we saw something blackish in the vial. I don't know what is that. We can't even examine it.
The thing is this, I would not even worry about what went wrong. I would trash it and just start new.
Okay, we'll
Yeah,
definitely do
yeah.
that.
Some problems in life are not worth investigating. If something went wrong, who cares what went wrong? Trash it, just start new. You can focus on a better problem.
Just borrow it from
Once my student had a bacterial this thing, so we were trying to get rid of the bacteria. So somebody told me that bacteria divides every 10 minutes and your cells divide every 24 hours. Do you really want to fight bacteria? No, because we can never win the game, right? No matter how many antibiotics we add, so we trashed it. So yeah, don't worry about these kind of trivial problems. Not worth your time. You're too smart for worrying about that black dot there, right? Next question.
What percentage of confluency do you prefer for immunoglobulin? Because
Not exceeding 60-70% under any circumstance.
50-60, the amount of protein collected is very less.
The thing is that when when proteomics people are working, they can make whatever confluency they want to make because they are just using it as a protein source. But if you are studying signaling things, right, you cannot go 80% to do that because every signaling is going to be messed up there. So you plate it on bigger plates or you use more plates, but you don't do that. So if you are really truly interested in knowing what is going on, your cells should always be at lesser confluency. And you know this thing that you cannot trypsinize it to harvest the cells for the experiments. You never can do that. You have to. You have to do it like this.
And
so we know that the epithelial to mesenchymal transition is necessary for metastasis to happen. So, are there points in cancer therapy where we trying to avoid that?
Oh,
blocking that
absolutely,
specific transition?
absolutely. EMT inhibitors. So now we know the players who lead to EMT. So we inhibit those players, EMT transition cannot occur. Absolutely, very hot area of research. Yeah. Cancer stem cell formation, stop it. Yeah. Tomorrow we are going to talk about pharmacogenomics. So what is that? Is that the same drug, why it has different effect on different people, right? So I asked you, a patient comes to you, right? So I will repeat it, a patient comes to you. So the first thing you see whether this person even has cancer. So you do PET scan first, and then you know the extent of cancer. You can also tell the grade of cancer, whether it is on lymph nodes, everything. So that gives you, okay, this is the cancer is there. Then the second thing is that you find out what kind of cancer it is, correct? And once you find out what kind of cancer you can have, then you can plan what kind of surgery or radiation or chemo or PDL1 or what inhibitor EGFR and all of these things, right? And then you have the therapy ready. Some things you can give without further testing, but for others you have to then do genomic testing of that human being, okay? And then, so for certain drugs it is known to have different effect on different people. So then you absolutely need to do this genomic testing, the DNA that we inherit from our parents, the germline ones. So they can do a mouth swab, right? Because it has nothing to do with the tumor. They can do a mouth swab and they can test it with PCR in one day, those few genes. And then you determine whether you will give the drug to that person or not. Because the drug to patient 1 may be very toxic. For patient B, it would be a miracle. For patient C, you have to give less dose. For patient D, you have to give higher dose. For patient E, something else. Do you see? So that will come in pharmacogenomics, why there is so much difference there. So that is something we'll see tomorrow. I think we'll stop for today, okay? Unless you guys have any question. Oh, absolutely. Ask me.
If a person is not having a cancer, but like how do they predict if like he or she will be going to have it in next 20 or
I don't think anyone can predict that part. I don't think anyone can predict that part, what will happen in 20 to 30 years. Okay, but if a person is smoking, it is easy prediction that person can get cancer. Although I know a lot of people who are chain smokers and they never got cancer, and they are in their 70s now. I don't know why. Okay, and so they have high potential to get cancer. If they are BRCA1, BRCA2 deficient, high potential to get cancer. Likelihood is high. So it is very, very hard to say.
I cannot recall, but I was reading something like genomic studies, they can predict the mutation rate and thus they can predict in the future like whether it will come or not.
Here is the thing. Suppose I eat a lot of antioxidants, I eat a lot of fruits, right? Suppose, hypothetically, and I take antioxidants and I do exercise, right? So you would predict that I should never get cancer, right? But you don't really know what are the factors, because this is the truth, that people who live a very healthy life, exercising this and that, they still get some disease, right? And why they get the disease, in many cases you can tell exactly why they got the disease, because something happened in their kidney and something was released and that had an effect and they didn't treat it and it became chronic and some inflammation was there which was not showing. So in every case you can find it out later why it happened, right? But the thing is that if that person was not exercising, maybe it would have manifested a lot earlier than later. So there are many factors. And I bet you didn't know that secondhand smoke is worse than smoking. So if you cross an area where you smell cigarette smell, even that is bad for you. And yesterday I met a lady who came as a chief guest in one of the conferences which are going on. And she was in Delhi at a very high post in defense. So we had dinner together yesterday. So I asked her, how was Delhi? Because she's from a different city which is extremely clean and nice. So I said, how was Delhi with all the pollution there? And you stayed 4 years. So she told me something really good. She said, when you're inside the problem, you don't see it. And this is true. When you're inside a problem in life, you never see it from outside. So she said, when I was in Delhi, I never saw the pollution because I was so in the problem. You know? So many times we are exposed to things we don't realize we are being exposed to, you know. So sometimes things can sneak in and we are not aware of. Sometimes we are chronically stressed without realizing we are chronically stressed. So yeah, it is a complicated thing. It's a complicated life.
What is the reason for the second-hand smoking is worse than the smoking itself?
I don't know that. I really don't know. If I have to, I mean, obviously we avoid smoke at all cost, right? I don't know, but if I have to guess, if a person is smoking, he's exposed to smoke while only he's smoking. But a person who is working in a bar is constantly exposed to smoking, like 24 hours a day. And the person who is smoking in his home or outside, 10 minutes he smokes and then he is doing something else. But the person who is working in a smoky environment is constantly inhaling that. Right? So I think that is the reason. Yeah.
Does the Saj number affects our study?
Oh, definitely. If the cells have gone through too many passages, then the cells will become more senescent-like. So you should, when you get new cell line, you should make many tubes and freeze them. And then after certain passages, you should trash them. It depends on the cell type.
Okay.
Yeah.